seat, but you don't need it.
Should I start?
Yeah.
Okay, so I had spent now 1 hour now to discuss one constraint now from biology, trial to trialability. I want to also cover some other constraints now from the data, and now I have to do it much faster than, because at least I have 4 different constraints. So we had heard a lot now about diversity of neurons and synapses, and this is something which kills at least the simple models for Boltzmann machine like sampling with spiky neurons there. So then also I think we need also to take really data into account nowadays when people can record from hundreds and thousands of neurons while the animal carries out a computation, and so this point to different types of neural network computation than is commonly considered. I want to discuss also some aspects of synaptic plasticity which haven't been discussed, and also that neural codes tend to drift at least in higher cortical areas, which also may change your perspective of neural coding versus neural computation then. And altogether, I think I view this, one can view this as constraints from the data, but you can also view this as clues. You want to discover how BNNs work, and so this gives you clues, what are essential features apparently of these computations in biological networks of neurons. So concerning diversity, we have seen a lot, and this is just one slide here from the Allen Institute database, where each line here reflects one measurement of a neuron then. So this is from mouse, this is from humans then. So for example, this is adaptation index, which measures how much the firing of a neuron slows down if you give it a constant input current. This is the average firing rate and some other membrane time constant. And you see about every neuron has a different profile in terms of this electrophysiological measurements then, right? And so this is something which again gives us a constraint, for example, when you want to reduce noise by duplicating neurons or subcomputations, right? Because duplicating may be difficult if you don't get the same kind of copy of a neuron again then. And I think for me, the particular challenge is also to take short-term plasticity of synapses into account. I think Anton mentioned this yesterday. This is also documented in the Allen Institute database. So this short-term plasticity should not be confused with long-term plasticity learning. It's simply an effect of synapses on the time scale below a second typically, which means that if you send the spike train to the spike train comes from the presynaptic neuron, and then if you look at the amplitudes of postsynaptic EPSPs, then they tend to change then when you have a sequence of spikes. Some of the synapses are facilitating, so they become larger, maybe even 3 or 4 times larger. Other ones depress, they become smaller, and then there are all kinds of mixtures of these two things. So in the literature on neurophysiology, typically one very often uses this model from Makram et al., where one uses 3 parameters, U, D, and F, to characterize this kind of short-term plasticity of synapses then. And when you look here in the 3-dimensional space, UDF, of this, each dot is one synapse there, one measured synapse, and you see hardly any 2 neurons have exactly the same short-term plasticity there. But you still see clusters of neurons, and so according at least to the findings of Makram et al., when you have a particular type of pre- and post-synaptic neuron, is typically nature has chosen a particular type of short-term plasticity. In particular, connections from and to inhibitory neurons tend to be facilitating. So when, for example, bursts come through, they become larger and larger, the outputs, whereas the connections between excited neurons, they tend to be depressing, which means it's very hard to do any computations in terms of firing rates, because if you want to communicate a big firing rate then after a while it simply shuts down the synapse, right? It no longer transmits this then. And so therefore it's a history dependence of a synapse, which is not taken into account usually by modelers. And I think an exciting open question is how does nature possibly benefit from this? And I think it forces us to think also in terms of computations, not in terms of inputs being vectors of numbers or values and outputs being another batch output, but really in terms of computations on time series. So this is just one cartoon one spike trend emitted by one neuron. It's sent it here to a facilitating synapse, and you see it goes up, and then it decays down, goes up. This is another to depressing synapse. So therefore, a neuron can actually then therefore send different kind of time series messages to other postsynaptic neurons then. And as I mentioned, I think strong implication of taking this into account is, I think, for theoreticians, computation, it becomes very hard now to take any artificial neural network computation or any kind of Boolean circuit or any circuit we like in computer science and implement it in one way or another with a model for BNN then. As soon as you put in this depressing synapses between excited neurons, probably this model no longer is going to work then, right? So therefore there's something wrong then. Either nature has chosen different architectures or this data are wrong then, right? But we cannot just really duplicate computation architectures from computer science given this effect then. And it also one other kind of effect is important because in many computation theories one wants to do subtraction. Say you have a predicted value and actual value, and you want to find the difference, right? And just this innocent computation is, I think, very hard to really model with more realistic neural networks, because we heard this kind of inhibitor neurons are not just mirror images of excited neurons. They have different firing probabilities. They have different connection patterns. And they have also different types of short-term plasticity of the synapses there. So therefore, you don't simply have an excited neuron. You can get an inhibitory clone, which kind of produces the same thing just with a negative sign as an output. I think this is really impossible to with any realistic neural models.
Seems to be missing one word. What comes after make it? Is it possible or impossible?
Make it impossible, yeah. So very good, yeah. So the most important word is left out, yeah. So make it impossible to implement subtraction, but no, my God, yeah. And so this, I think, was really for me kind of a point to really to pause and think right now that we really have to maybe rethink computational design then if you take these effects of biological networks seriously. And I think it's a good chances that the biological neural networks simply have chosen quite different computation architectures, and we should discover them, right? We should not always reproduce clones of ANN architectures, but really discover new types of doing computations which are supported by these types of data then. And I want to a little bit also, yeah?
Could it be that the variations in the synaptic responses are averaged out because due to lots of synapses impinging on a neuron?
I think this would, one aspect that speaks against this, as I mentioned, typically if you have a population, say, of excitatory presynaptic neurons and a particular type of, say, excitatory postsynaptic neurons, then the synapses tend to have all the same type of short-term plasticity. So although it changes somewhat then in prefrontal cortex, it's apparently more facilitatory. In other cortices, it tends to be more depressing than. So it's not a mixture of depressing and facilitating going from the same pool of pre- and postsynaptic neurons. So therefore, I think this solution may not work out. Actually, so to mention, I think this has been reported also by Mansfelder from Amsterdam, also for human neurons, so that they're depressing, they're strongly depressing neurons, synapses between pyramidal cells in the human brain. Yeah.
So what aspect of having short-term plasticity makes it impossible to implement subtraction in a single unit?
Well, no, you have here a neuron A in an artificial network neuron B. You want to transmit a message, a firing rate, right? So now you do it now with real synapse, and so therefore the amplitude of the postsynaptic EPSP becomes smaller and smaller for later spikes in this spike train. So if you want to use this rate to represent the variable, you don't have any invariance, right? So in some sense, synapse can no longer be characterized by a single parameter. It has no temporal aspect, and you have to this history dependence of the synapse has to be taken into account then. Otherwise, you have huge kind of deviations from how this network should behave then.
So is subtraction not the arithmetical operation of subtraction that we think of it?
This is another thing. I think one thing is rate coding is not possible. It becomes difficult, but also kind of really producing kind of the negative of a result, kind of an inhibitory clone of excitatory neurons, I think is also impossible. I wouldn't know really how to put it together with any neurons and synapse types which are in the Allen Institute database. So.
One way to think about that is that a code is the asymmetry that you only transit positive numbers, right? The neural code cannot go negative. You either have 0 or positive values. So you can't transit negative remainders.
Yeah. I think one really has to think also in the end about neural coding. For example, I think one idea is simply that excited neurons, each of them is an expert. Whenever they fire, they want to give a particular message, they convey content, right? And then there are these inhibitory networks which want to make sure that not all the time all these neurons want to kind of raise their hand and say it's my content, right? So therefore it's more regulation of experts. I think it's closer to the way how biology uses excitatory neuron and inhibitory neurons, rather than the way how we use in artificial neural networks, where just inhibition is just a negative of an excitation then. So I think one question I want to quickly run through is whether what are neural models which are, or network models or computation models which can A, be compatible with this diversity, but B, also can possibly even benefit from this. And I think now, as I mentioned also, I think if one really wants to understand biological neural computation, one should think in terms of time series there, of network states then. And I think one of the nicest early results was when there was in the cricket, I think in the Gillerong lab then, in the neuron paper, they gave 2 different odors to this animal. And so then they recorded, I think from 87 neurons simultaneously. And so these are 3 PCAs of this high-dimensional network activity, and see it moves out here and then it moves back then. And when they give a different order, it moves out here and then it comes back. What's missing is that this trajectory of network state, it never moves to a point to an attractor, which is then by theoreticians postulated as being the code for this particular order, right? It keeps moving all the time then. And so this seems to be something which is quite generic then. Whenever you look, then you hardly ever find that a network state stays fixed, really put there in an attractor like basin or so. They always keep moving. And so therefore I think one has to really entertain this idea that when you look at the dynamical system as a model for brain networks, it has attractors and it has this trajectories towards attractors, that in the end these attractors are very transient, and most of the time the network is in these trajectories and really computes in terms of trajectories. It doesn't compute in terms of fixed points then. And so therefore this is a different way of really using a dynamical system for computation then. Yeah.
What are the advantages of that, do you think?
I don't know. I think it would be an advantage if one would postulate that an organism really gets as input always a time series. An organism never gets a batch input, and always has to really produce an output series. And it's artificial when you have to make one output decision, or when your animal has to decide all the time flee or approach then, right, to forage or not then. So you have both on the input side and the output side as streams then, right? And I think one should think of the computational engine being a filter really, which operates, transform an input time stream in a target output time stream then, but it's really on time series. So therefore, I think one can entertain that this is just the computational context of the brains then. And doing multiplication or so is really a very artificial, contrived a computational task then.
But if the organism is ultimately to make a decision about its behavior, it needs some kind of metastate that's derived from that trajectory. It has to know what trajectory that system is on.
It's a good point now. I think if I come to the slides, we see, but also one sees in the data from Mike Shedlin that there are special neurons which integrate over seconds evidence for or against a particular decision, right? You have move left or right, and you see competing neurons which each integrate evidence while the rest of the network is quite active then, right? So therefore one sees also there are methods apparently how the brain can transform a trajectory of states into, in the end, a decision at some time point then. Yeah.
Well, just in support of what you said, invariant, you don't want it stuck on one set of neurons. So you get tremendous protection if you put it on some sort of trajectory, so that if pieces, all those places have to be calibrated. So if it's resident in one particular area, that puts a lot of stress on the model to make that happen. Whether it's just floating, then it takes the heat off those issues.
So I think that maybe you're arguing that there's some kind of simply system level benefit for having kind of representation moving around rather than being stuck there where these ones fire for a long time. Yeah. So I'm not sure whether I should discuss it now. You can look it up in the slides. I pulled out this slide here. It's a result from Leon Chua, who is here in the faculty of ESCS, had proven this very nice theorem. And it tells us, I think it's one of the very few theoretical results which says there is a benefit in having diversity of computational units. Then I think almost all results I know is saying things get worse if you have more diverse units. Then so this is one result which suggests, in a very constrained setup, it's good to have diversity of units. Then so here let's quickly go through this then, so it's about approximating a given time invariant filter with fading memory. So this could be also equivalent to filter represented by Volterra series there. So not only linear filters, but nonlinear filters. So you can approximate any such filter with having two types of units. Here, this is a memoryless output unit. Simply, kind of think about this as a kind of artificial neural networks feed forward, without any temporal operation. But then here, it's enough to have in parallel a filter bank where each of them is a filter and altogether this ensemble has to satisfy this pointwise separation property. And so this is a demand on the diversity of the selection of filters, because it says when you have any two input functions on which this filter has to operate, which used to be different at some point in the past, there has to be some filter in this pool which gives a different output for this two function at the current time step. And obviously, if you wouldn't have this property, this couldn't give different outputs for U and V then, right? Because this is here a bottleneck for the information about the input time series going to the output. And so it turns out that this pointwise separation property gives you sufficient property for all this temporal computation. So one sees one can in principle piece together quite complex nonlinear filters if you just have a rich enough filter bank then, right? I don't want to propose that the biological system has in parallel filter bank, right? But think in terms of recurrent neural networks, maybe similar effects are used. And I mainly give this, want to show this here because I think there's an open problem, really a deep open problem. I think also for theoreticians, particularly computer science theoreticians, is can we provide further theoretical insight into having the computational benefits of having diverse computational units within a network. This is not a great example, but I think it's one of the best I have found, or the best even. This is, I think, it's a nice open problem. So let's leave it there then. There's this theory of liquid computing, which was mentioned, which makes use of these things. You can argue, but maybe I leave out all these things then. Yeah.
So can you give some idea about the proof of the previous theorem?
I don't know how it's proven. Yeah. No. So. Yeah. I think I don't want to go into all of this then. Yeah, maybe I think I mentioned only about this liquid computing. Idea is that this is one way of just doing computations when you have you can take even off the shelf a network which is constructed according to the Allen data database, also with having diverse neurons, very complex connectivity, having synapses with different short-term plasticity. If you put enough of them in there, and then you train a linear readout from these complex circuits to do a particular computation, this works typically very well then. And so this is probably the reason why this has become such a popular model, because it's one of the few things that works even for spiking neurons, even for very weird types of neural models then. And so I think I don't want to go further into this then. And I think the only theory that I know of in the back which supports this is this theorem of Boyd and Chua, which gives a little bit, at least goes in the direction of supporting kind of benefit of having diverse units then. So then there's also now, here I have some slides about what happens if you add now feedback from the readout back in the neuron, and then suddenly the theoretical computational power of this type of network really jumps then. So suddenly again, you're no longer stuck at filters with a fading memory, but you can even simulate any dynamical system formulated in terms of a system of differential equation, a special case also any Turing machine. So this is something where a lot of recent work also looked into this question now. When you train now a readout, which not just goes to downstream networks, but really project the readout value back into the network, then suddenly you can also practically carry out very strong computations then. And so this is at the moment quite a bit of debate then, what are good learning algorithms for doing this then. And I think I have a slide now, which discusses this recent literature, so you may want to look it up. So they're all about different ways of training these readouts with feedback then. So there's this force algorithm which was proposed by Susilo and Abbott a while ago. Just very recently, Claudia Koppert has shown that this algorithm can be applied also to networks of spiky neurons. And really, it's just practically, it's a method of generating non-trivial computations in these networks. Then I think the point where some work needs to be done is that I think none of these authors would argue this force algorithm is biologically realistic. So if neural networks or brain networks use such a strategy, they use also different learning algorithms. And so far one has found, if one has looks at this in the context of artificial neural networks, one can also do this training of readouts with reward modulated Hebbian learning, which is reasonable, realistic from biological perspective. It's an open question whether also for spiky neural networks, the force algorithm for training readouts with feedback can be replaced by biologically more realistic learning methods there. And a kind of a technical additional challenge for all these learning algorithms is always, since you have this feedback, this learning has to home in very fast to good solution. Otherwise, it's simply, because it projects its errors back into the network, it simply gets stuck in bad solutions then. So here now it's really key that the learning algorithm converges extremely fast to good solution then. And then there's a number of experimental data which have in principle support predictions of this type of liquid computing with diverse units then. So the fading memory property has been shown. Then there's also this idea of multiplexing of neural codes is something which grew out of these ideas, and this has been supported by a number of exciting experimental studies. And also this effect that in principle you can have a diversity of different readouts which all get input from the same cortical column but really do different computations. This also has been shown to in some one special case to take place in cortical columns there. I want to move here to two other points then rather because I think they are more newer than and maybe also have more open questions associated to them. And so one is that really a genuine difference between what theory predicted and what should be the typical network activity for network of spiky neurons which carries out computation. So this has been postulated that this should be in the so-called AI regime, which is the asynchronous irregular firing regime then, which would be here. These are about 400 neurons. And so this looks like white noise then. And if you add up the rates, the sum of rates, it looks like this then. And this was kind of considered as the most beneficial regime for a recurrent network of spiky neurons to carry out computations, because the other ones had kind of oscillations or other artifacts which were bad then. But now, if you look now at data, when you have recordings from many neurons at once, so these are from electrode recordings from the Harris lab, then from A1, area A1 in the rodent, then you see this is time here, 1 second. So both spontaneously and when tone comes on, you see that there's certain kind of population events then. They call these typically assemblies of neurons then. And then also what they pointed out in this study here is that these population events, both before there's any auditory input and when auditory inputs comes, they're very similar to each other then. Which is, I think, for me, looks like the worst type of neural coding you can choose for. A, you don't let each neuron code something independently, but also you have this network activity stimulated by these assembly activities, and you find the same assemblies when there's no tone, and the same is almost the same assemblies on average if there's a tone. So this is here one line for each neuron, and the red dot marks here on the time axis its preferred firing time on repeated activation of this assembly then, or you call it assembly sequence. So there's always a little bit of time course. In this case, rather fast time course there. And I think what we are missing is now a computation model which can A, live with this type of neural coding, and B, can benefit from this type of neural coding. And I mentioned already that possibly if one thinks about also stochastic computation, if a certain assembly goes on, maybe one could view this as sampling that the local network states switches now to 1 or so while this assembly is going on then. So this could be just one possible interpretation. I want to mention also the same thing has been found now also taking place in area V1 then. Even when natural movies are shown then, both spontaneously and during movies, you see very similar population events. So this was used to lab with calcium imaging. So therefore, I think we really have to kind of face the fact that what theory proposes as being the best activity regime of spiking neural networks is not the one nature has chosen then, right? And I think we have to retool really and readdress this existing data then. And I'm somewhat alarmed by the fact that these are kind of old data, but I think when I read theory papers, they still not talk about this AI regime then, right? So I think there's a very slow kind of transmission of information from experimental neuroscience to theoretical neuroscience. And I think we have to shorten this transmission time from something like 10 years to 1 year or less then. Otherwise, we're not making enough progress then. So here I have also kind of put together a number of references if you want to learn about these aspects. There are a number of very nice papers which all of them address both actually experimental data of this assembly activity, but also some ideas about the computational implications from them. So this particular, this is a very nice, it's a brand new science paper where they proposed that assembly coding affords is a certain very clever hashing process which has a computational implication. There are other types of data and reviews which address the fact that possibly memory contents are also encoded in the human brain by assemblies of neurons, and perhaps Christoph Papadimitriou will discuss a little bit also from the experimental perspective. And so I think there in a rough model, we would say also if you think about a banana, then probably an assembly of neuron gets active in your brain then. And so therefore, kind of higher level thinking apparently takes place in terms of activation and deactivation of assemblies. And what we are missing is really a computational calculus which supports this type of computation with these types of encoding there. So let me also now talk about a little bit of unexpected properties of synaptic plasticity, where we have actually talked very little, or the talks were not really addressing much synaptic plasticity. I find this actually for theory and modeling kind of the most annoying kind of lack of knowledge. That there's a huge literature of synaptic plasticity, but if you take a close look, there's a large diversity of results then. And whereas theory usually uses one learning rule and just says this is universally used in the brain, then whereas the data don't really support this. But the additional effects, we have already heard that in principle there are the spines on which kind of axons form synapses, and now as soon as one started to be able to record from the same dendrites with calcium imaging over many days, one found that besides spines that are kind of there all the time during experiment, there's some which only there for 3 days and go again. Another spine was only present for 1 day. And this is something which is apparently inherent properties of spine, because the kind of growth and shrinking of synapses is according to this data also takes place if there's no neural activity. So it's a type of changes in synaptic strength then, because spine volume is a good proxy for synaptic weight then. And this takes place even when here spiking activity was stopped here at this time point, then it even becomes somewhat larger there. So it appears to be a genuine molecular encoded property of spines that they all the time kind of have some dynamics going on then. And so this is apparently one large factor in synaptic plasticity. One other aspect of kind of unexpected property of synaptic plasticity really emerged from two data sets. One data set is really this data set from the Lichtman lab, which Jeff discussed yesterday. They had some lucky cases where they had here this green dendrite and then here this blue axon, which formed here several synapses on the same dendrite. I think here even 4 then. Now, when you look at any learning rule, you see all these synapses here, these yellow synapses, they have the same presynaptic neuron, they have the same postsynaptic neuron, they even have the same postsynaptic dendrite, right? And it's very close together. I think this is probably 50 microns or something of this form or less than. And so then they looked at kind of what is the correlation in the strengths of these synapses in terms of, in their data, in terms of spine volumes. Another data set from culture from the SIP lab, they looked at sizes of PSDs, postsynaptic densities, and they found the correlation between the synapses is there, but it's pretty weak then, right? No, it's 0.23 then. Whereas I think the common thinking about synaptic plasticity as a goal-directed learning would suggest the synapses have to have all identical values except for differences in initial state then, right? And so this, I think, is a puzzle there. This weak correlation in the strengths of synapses which have the same pre- and postsynaptic neurons there. And so this is one thing, so we asked now, could it also be indication for a different strategy of synaptic plasticity and possibly even have benefits? And so this is one theoretical framework that I want to quickly go through now as the last component of my talk then. We recently also looked at some data from the lab of SIPs. So these are cultured, but there has advantage. They can image over 48 hours the sizes of postsynaptic densities, or more specifically the number of PSD95 molecules. And so these are now here pairs of synapses which have the same pre- and postsynaptic neurons. And you see that not only when you take a snapshot, you find different values, but even if you normalize them to 1 at the beginning, they drift apart then during these 48 hours there. And so this supports this kind of hypothesis or conclusions of these experimental papers that synaptic plasticity is to a large extent, here the blue area is driven by spontaneous synapse autonomous processes. And these are those components of synaptic plasticity which may be run really by what we usually call learning algorithms there. So this is a second level of plasticity, right? It can contribute also to trial-to-trial variability, but this is a level of stochasticity which probably takes place on a slower time scale then, right? Rather than this firing plasticity and synapses which don't release, which is noise on a fast time scale. This seems to be noise on a slower time scale. And again, the question is, what could it be good for?
Wolfgang, there's a study, I'm not sure if it's that one. Is Kristen Harris one of the authors on this one?
There was one where Zinovsky was one author which also studied this and arrived at a different conclusion then.
That's right. They were sort of claiming and with great excitement that
it's the same,
there were 7 bits.
So this is something I discussed this with Noam Sif then, and so it's discussed in their paper, which was in PLOS Biology in 2016. So they argue that they had used a much smaller data set than of, I think, 17 or so of these pairs then, and also they had ignored some of the data then. So this is a debate among neuroscientists, yeah.
Yes, sort of almost the opposite.
But I think the strongest data set comes from Lichtman. I think he had, it's in the supplement of their 2015 paper. I think it's 100 or so of such pairs then of synapses which have the same pre- and postsynaptic neurons there. And so this is, I think, apparently a good data set. But I agree, it's really on the cutting edge of technology right now, these types of measurements, and we have to look out for this. So
why do you expect a high correlation between?
Well, this is what, as a bloody theoretician, I would say, they have the same learning rule, right? And so if they start with the same initial state, they should always go in lockstep, like a ballet.
But the ultimate measure here is the strength of the connection between those two neurons. And so you could just look at the sum of of those 4 synapses.
Yeah, right. Well, but then you would,
Having to have them
okay, this would be good, yeah. But then we have to think about now a new type of plasticity algorithm, right, which organizes all these 5 to 10 synaptic connections between 2 neurons, right, and the communication between these 5 to 10 synapses, which are typically apparently not on the same dendritic branch then, right? So it becomes a non-local, non-trivial learning rule, but certainly could also take place, yeah?
Not necessarily. Couldn't you just have a probability for each of the synapses to learn at some instant? Like, you'll have like the added
Right.
and they'll learn separately.
Right. No, it's exactly, I think, what you suggested. I think it suggests to look at probabilistic learning rules then, right, rather than deterministic one. Every learning rule I see in books in neuroscience or papers is a deterministic rule, right? Delta W and then something deterministic there. And so I think to me this suggests to look exactly at probabilistic learning rules.
I know many of the learning rules, well, STDP in particular, they do it for like 50 paired spike trains or something like that. And so at that point, like, you get a very clean distribution, but it could still be a very probabilistic process at the base.
Right, and sometimes I think you agree now. I think now, if you look at the SDP data, typically you have a nice SDP curve and lots of dots, right? And each dot is one experiment, and they're all over the place then, right? So each synapse plays a different kind of role then somewhat. Let me also move on here to something else then, which I think is for those of you who love neural coding as a firm foundation for neural computation. So I think all the measurements which have really tried to track neural codes over a longer time span, and apparently the CIF lab at Stanford was the first one, they found that they drift on the time scale of days to weeks then. So this is here when this rodent runs through linear track then. So on day 1 you sort them according to firing times, and when they run again through the same track on 5 days later, you no longer find this, right? You find a new kind of linear relationship in the firing probability if you reorder them according to the firing time at day 20 when they run through the same track then. So apparently these place cells are shifting then. They argue you can still kind of extract some invariant knowledge, but the place code, they argue, is something which is drifting. And more recently, this paper from the Harvey lab appeared. This is in posterior parietal cortex of mice then, and they have also kind of an assembly sequence encoding a kind of a learned two forced choice behavior. And they also argue that on the time scale of 10 days, this quite a bit changes this neural code then. And so therefore, I think this data suggests that neural coding, at least in higher parts of the brain, is not a firm foundation, but it's drifting, right? And so this is something which also should give us to think, because how can you have invariant kind of control and memory and being able to walk, right? When things are drifting in your brain, we need to look at theories which can live with such drifts then.
Oh my goodness. The hippocampus is supposed to be involved in maintaining, it's an episodic memory that goes back in time. Excuse me, I don't know, something weird happened in my lip. And in a rodent, that's supposed to be, the number that sticks in my head is like 30 days. After 30 days, the information that was trapped there episodically during, is gone. In a human, it's about a year. And so you're calling it a drifting representation. Maybe it's a representation being forgotten.
Yeah. Actually, I'm not calling this is what the author called this then, yeah.
Right, I'm just making it.
Yeah, but I think, well, on the other hand, these are not memories, right? This I think something different. The rodent runs through the same linear track then. So it's not memorizing something or recalling something, really running through the same linear track then. And so I think one has to really, this neuron here kind of fires for a particular place on this first day, and it no longer fires for this place on this day then. So the place coding of neurons on area CR1 is changing according to the data. So right. I think it doesn't have, at this point, something to do with memories or episodic memory, but with place coding.
Okay, I would say remembering to know to fire at that place, it's storing something, and then if it no longer responds to it later, you could say it's no longer. Anyway.
But maybe let me also a little bit go ahead in my talk then. So I think coming to a few open questions then. So I think I think this is for me, I think one of the most pressing problems, particularly also for theory, also kind of from computer science. How can BNNs attain and maintain stable computational function if everything is changing, then also on slower time scales, right? Neural codes are drifting, probably synaptic connections are drifting, synaptic weights are drifting, even if there is no overt learning then. So another question, is there anything invariant? And so I want to here sketch a theory which in principles allows us to think about something which remains invariant there. And so we are looking at the situation which was here suggested by this young man, that we look now at learning rules which have a stochastic term. So here this is now a learning rule. I'll tell you in a moment what theta I is. But it has here kind of an infinitesimal step of a random walk. And so this is now a stochastic differential equation, no longer a differential equation or difference equation as one usually uses that to write down plasticity rules. And it has here a drift term, which I discuss in a moment, and here a diffusion term where you have a temperature parameter which scales how strong is this kind of random walk aspect of the synaptic plasticity. The way in this model, how this parameter theta I was chosen, it was chosen so that it both kind of controls synaptic connectivity, because it's used in this model so that whenever theta I is larger than 0, then this encodes a functional synaptic connection. And in this case, the exponent of this is then the actual synaptic weights then. So this exponent transformation was just chosen here because then one can write down simple equations and fitting them to data about this dynamics. So here the goal is to integrate rewiring and synaptic plasticity, change of synaptic strength, into a common theoretical framework and learning rule. Now this looks like an awful equation here. So by here the way I have plugged in something which is a black box. In principle, you can plug in here any distribution over network states. So theta is the vector of all this parameter theta I in a network. And here I have a partial derivative with regard to the log of some given p star then. Now the Fokker-Planck equation tells us that if all these different stochastic equations for each each synaptic connection are running simultaneously, we can say something about the station distribution of this network now in terms of parameters, no longer in terms of network states, but parameter vectors. Namely, it has as a unique invariant station distribution something which is very close to p star. It's modulated by 1 over t then. So therefore, if high temperature, it's flattened then. If low temperature, the peaks come out more strongly then. And so therefore, with this type of learning rule, you can in principle use this now as a new scheme for programming a desired behavior of network configurations into the network, where particular network configurations are favored then. And I want to discuss here the case where those network configurations are favored, where this network gets large rewards then. And maybe also some prior plays a role here, which expresses you want to have sparse activity, for example, or kind of genetically encoded knowledge. Really, this could be the connectome already, that certain synaptic connections should be there, if not at all, overruled by something else then. And so then it turns out if one really plugs this into this very theoretical ansatz in here, one sees that then if you look at the partial derivative which arises here, one arrives at in some sense well-known rule for reward-gated SCDP. Then here you have eligibility trace whenever, say, an SCDP event took place, then it decays over time, and R of t is a reward then. So this means whenever a reward comes, recent SCDP kind of events get kind of engraved then. But you have, as a new aspect here, you have this stochastic term on the learning rule. And so therefore this produces overall a completely different picture of this learning dynamics. You no longer can think of a network of starting somewhere and moving to a good configuration and staying there. It will keep moving all around then, right? The network keeps sampling then, even if there's nothing to be learned then overtly. Yeah?
But I imagine part of the learning makes the distribution on a stationary, right?
Part of the learning?
Yeah, so I mean, it would be hard to tease out. I understand that, during this diffusion, you may end up, you will go to a stationary distribution, but also learning could make the distribution change over time. So there is that.
I'm not sure, but I think here learning is kind of kind of doing the sampling here. There could be a meta-learning taking place that maybe after a while, like when you grow older, you suddenly change your goals, right? You no longer care so much about money, but whatever or so, right? And so this would modify on the larger time scale of meta-learning, possibly your state distribution. But here, the learning is part of the sampling here, right? This is a learning kind of rule then in this model then. So let me also know, I think rewards, dopamine is supposed to do this. And so we applied this as one test to very nice data from Nature paper 2014 from the Kumiyama lab, where they had a mouse, where they monitored many neurons there. And this was trained to do a complex lever pressing task. They first had to push it down and then pull it up, and then they got a drop of liquid then. And so they needed quite a number of sessions to learn this. So this is kind of checking here this movement over time, and you see after a while, session 14, it becomes relatively stereotypical. And also they checked here the neurons activity. So you see assembly sequences emerging them and becoming cleaner then during learning then. So the neuron fires, most of them fire only as a part of this assembly sequence. So this is consistent also with this data from Chris Harvey from postparietal cortex that apparently learned behaviors are encoded in higher cortical areas like parietal cortex and prefrontal cortex by assembly sequences then. And also they are reported here ongoing spine activity, kind of new spines coming on early when the learning started and later again elimination. So this adds to this pruning aspect which Jeff yesterday mentioned also that when a new learning task arises, then the new spines, new synaptic connections are tried out, whether they will be useful then. So this is something we simulated in a model, and so this is now a model which I think in some sense is nice, as a discussion point with regard to all this results about diversity or so, because we are now working in a regime where you simply can put down here a model which is as diverse as you want to, or realistic as you want to. You can simply take off-the-shelf network based on data from the Allen Institute then. And so here, the only rule that was, or the only thing that was putting structure into this was this learning rule, which is the one reward-based learning rule which I sketched then here. And so here we kind of try to mimic in a simple network model somewhat similar learning tasks. So here an input neuron simply gave a prompt then, and then there were two populations of neurons, and activity in this population kind of pushes the lever, symbolic lever down, and activity in this pushes lever up then. And so then when you start training this network, it gets a reward whenever it does this right, first pushing down and then up, then it gets a reward here. So he indicated by red. And after several hours of learning, it learned this to do this quite efficiently and fast then, so with occasionally kind of failures or so. So therefore one sees in some sense this reward-based learning in this stochastic context can reproduce on the level of emergent network computation these results which were recorded then, and one can also see similar assembly sequences emergent. I want to address here also one additional aspect which now comes back to the stochastic nature of the neural code, or also synaptic weights then. So here we checked in the first three principal components the whole network vector theta. So this is the vector of, actually technically not all, but a large fraction of this network parameters theta I, so synaptic weights plus synaptic connectivity then. And so at the same time during this trajectory, kind of the performance level was plotted. So when learning starts, performance completion time for this two lever pressing task is long then, it's blue. After about 4 hours, it becomes red then. And then it in principle has learned the task, but the network keeps drifting around here. So there are irrelevant dimensions apparently of this network connectivity and plasticity which don't care for the task. And here we did something devious. We changed simply the role of these two populations. So now activating here now suddenly pushes the lever up and vice versa there. So it had to completely relearn then. But what happens here then in this, which is an inherent aspect of the model, you don't have to tell the model now to start learning again. It will automatically simply keep sampling then. And now those network configurations, theta, which give high rewards for this new setup, are the ones where it's going to linger then longer then, right? So therefore, kind of compensation for changing in the network or the task then is automatically performed by this. You don't have to put there a separate kind of loop on top of a learning algorithm which kind of says, now we have to relearn then, right? So this is something inherent in this then. And one aspect which is also nice then, this is at least qualitatively, it's consistent with this experimental data which I mentioned then on the strong fraction of the spontaneous synapse autonomous process, which apparently, usually, sorry about this, I have to learn. So which we usually we can't really interpret from the perspective of synaptic plasticity or learning then, because if you look at for which temperatures this network performs best, including learning fast and also relearning after this drastic switch in the network, we found that this works best for intermediate temperature, which produce also a similar amount of synapse autonomous dynamics there, meaning where this T is sufficiently large to also produce a lot of kind of autonomous plasticity. So this can be seen as a first weak kind of link between this experimental finding that synapses have their ongoing internal stochastic dynamics, and the model which puts it in the proposed functional use then, because it enables the network to keep sampling always from new network configurations, to be opportunistic to see whether they're better ones, or particularly also if it's disturbed, to come to a new kind of better solution then. So lifelong learning in a sense is supported by this. But at the same time, I think also this simple model kind of gives room for the type of ideas that Jeff Lichtman yesterday discussed. He discussed a lot his thinking that the connectome really contains a lot of knowledge which nature has accumulated during evolution then. And so particularly here one finds, this was a relatively small network, I think of 50 neurons or so, and it took hours of simulated biological time to learn then, right? So possibly one kind of way how nature can enhance this is that for really occurring learning tasks, the connectome enables the network to faster go through network configurations which are likely to be good for learning something that is important there, right? So therefore here, you can of course look at this from a complete tabula rasa framework, everything can be learned, but then nature doesn't really kind of support organisms which can everything learn sometime, but fast learning is a key then, right? And I think a critical or interesting question now for theory is to look at kind of connectivities which together with such learning models allow you fast learning and relearning for a limited range of computational tasks. And so this would give a new way of understanding the role of the connectome, not as something that is fixed, but really giving a kind of a base level of network activity and learning capability, which allows you then to explore variations around this base level of network connectivity. Yeah, then in principle also as you probably have noticed, this talking about temperature, we are here very close to very powerful optimization method, simulated annealing, which guarantees that in principle, if one decreases the temperature according to a student sufficiently slowly, then this network has even a theoretical guarantee to finding a globally optimal solution. So this is, I think, an interesting question, whether nature uses such cooling methods then. So let me the last 5 minutes maybe talk about methodological issues. And I think we have seen from having all these wonderful talks about which were driven by biological data, and now here these considerations which look at this data more from the perspective of getting network function models out of this. It's a question of how to choose a model then. And I think we should become more systematic about this. And in principle, you all know it's good to choose simple models, right? Sorry about this. So Newton was, I think, very good. He ignored that there's friction also in the air then, right? And then he could use evidence for kind of the laws of gravity. But assuming Newton would have decided now to understand the trajectories of leaves which are kind of moving around then, right? So now it's a question then, should he also say, well, wind is not, I can ignore wind, right? No, I want to have simple models. It's just gravity, right? And I think this is the question we are in then when we look at biological data and also function of these models, right? Now, computation function, what aspects of these models that we see are kind of here essential for this particular task that we are aiming at, and which ones can be selected. And if one simply looks at literature, I think there's a very strong bias towards one particular way of regularizing models for BNNs. Then one tends to use the mathematically simplest neuron and synapse models. And I think this is a tradition which is kind of very well justified, simply from tradition of mathematics and theoretical computer science, right? You want to have simple, clear models, and then you also can have the nicest mathematics. But I think when we really want to understand BNNs, I think we need to kind of consider also other ways of regularizing. I think we need one way of kind of weeding out models and avoiding that we have too many models, otherwise we're overfitting, right? So we should in the end have also as small as possible pool of candidate models. But I think, for example, you could also use kind of as one regularization methods that you want to focus on models that exhibit a similar robustness to network perturbations as biological neural networks, or use models that can learn fast, then or also use models that are consistent with the more experimental data about biological neural networks. So one sees, if one really stands back and asks what are the options for regularization, there are many different ones. And I think we have to kind of discuss these options more kind of more consciously and more systematically. I think typically when I see a paper in neuroscience, they have a simple model and they say something is biologically plausible. This is all the discussion about all of this. I think it needs more than one sentence in, right? Now we simply look at the pros and cons of choosing different models, simply looking also at the options for modeling then. And I think it would be very nice if here also during this coming weeks, we can discuss somewhat what are the right types of models, what are the options. And in the end, I think it becomes more a sign of mature science if we have also this methodological discussion going on and we can justify why we use this model for this task. Then we all know there's no ideal solution. All models are wrong then, right? We have to ask for what can I expect from this model? What are the risks of really coming up with completely biologically irrelevant solutions in this model, right? So it has to be more qualitative discussion then. There's also kind of another kind of principled method for at least classifying models was already a long time ago proposed by Maass and Poggio, so that you have the abstract computation level, then the algorithmic level, then the implementation level. And there's a very long tradition of theoreticians of saying, let's just think about this, then what needs to be computed and what are good algorithms for this, because there I can use all the tools I'm familiar with, and then somehow somebody else has to figure out how can biological neural networks really implement this strategy. I think what my impression is that now we have so many data really about BNNs that we have to take this seriously, and I think I've tried to show in my lectures that simply taking this data seriously suggests particular algorithmic approaches, and in the end also different computational kind of approaches like sampling or so, which otherwise may not be suggested, if not suggested by biological data. So I think we have to learn also to take all the three levels of this Maass hierarchy seriously. Yeah, in a recent paper together with Christos, which emerged simply as a preparation workshop for this Brain 2018, we had also tried to collect a number of references and also open problems, which particular could be of interest for computer scientists who look into understanding brain computation. And I want to give reference to this then. So my summary is that my personal hunch is that the organization of computations in BNNs is very different from schemes that are familiar from computer science and artificial neural networks. There is something to be discovered there. they discussed also the impact of a few aspects of biology, and they make specific suggestions also for computational organization. There's another set of kind of suggestions which emerges from what Bartlett discussed then, right, which I didn't touch upon at all then, which again I think suggests different types of organization of computation and learning also. This is all, yeah.
I want to ask you if you can elaborate more on the nature of p-theta on your Fokker plot, because I find it very intriguing. So basically, if I do like a one-shot learning, the p-theta, what would happen to that?
If I do a bunch of learning?
One-shot learning.
One-shot learning.
And also,
I think I'm not sure whether it really cares about this, right? Now, this is here about a kind of first you would have to formulate one-shot learning as a stochastic differential equation. If you don't have a stochastic term, you can't apply the Fokker-Planck equation, right? It really allows you only to analyze stochastic motion, like now was apparently invented to analyze Brownian motion, right? Where it's very hard to predict the trajectory of a particular particle, but you can say what is the probability that there are many particles over here then, right? So if you look at this, step back then and look at this evolution of the distribution of network configurations, then you find laws then. And it certainly, I think it doesn't give us all the information we want to, and I think you're driving at I think a weak point of this analysis, because it doesn't tell you how fast it converges to the station distribution, right? This is what you would need for fast learning typically. And so therefore, again, now you can productively maybe turn it around. So therefore, we shouldn't look at the most general network, right, and the most general learning rules, but ask, how can you support fast learning of really important tasks or so, rather than being a completely general then.
There are techniques that take curvature of these, information geometry techniques that make this much faster. But in general, I think my question is about the framework. You're really envisioning a single distribution for the brain, basically.
No, I wouldn't go so far. No, look, I'm trying to look at models for brain computation and learning. No, I don't want to say the brain is like this then, right? I think at least I feel we're much further back then, right? At the moment, I think we don't understand a single thing about brain networks, right? So I would be happy if really producing at least something which moves us in direction of understanding computation in brain networks. But it's a long way off then. I think one should be realize how far we're off. And for me, one criterion is actually looking at, Jeff yesterday suggested this scientific U then, right? We get this data, and we should also reproduce this. So for me, I think when we are able to reproduce really rich computational and learning capability in constructed networks of spiky neurons, then we have understood something, how to work with these devices then, right? But I think, as far as I know, we are far away from this, right? So we have a lot of papers which argue, this fits to this data. But if you look at data, typically experimental data are low dimensional. So every kind of model with many parameters can be fitted to low dimensional kind of constraints then, right? So I think the evidence for current models being really close to what the brain does is not so strong in my view then. And then also diversity of brain computation and brain areas. So I think there are several, behind you.
Yeah, just curious for some context. In some of these artificial spiking neural networks, how many neurons are there in some of these experiments?
Yeah, so here this were few neurons, I think 50 or so, and so this on our smallish cluster it was just enough for this then. So I think when scales this up to a few hundred neurons, one needs already a pretty large computer then to carry out this. So I think that we're kind of getting close to a situation where at Google or so where you have large computing capabilities, you can explore many of these models much better than we can do with current smaller simulations. And yeah.
So my question is, so I think your lectures gave a very good picture of really what in neuroscience what the observational data are and how they are really challenging our models, like the drift, like the hippocampus 10 days later, the code looks different and things like that. But on one of your slides, there was also this statement, for example, learn from few examples. And so I wonder how much should we take constraints seriously that we know about behavior and what brains can do. And for example, to take this seriously, learning from few examples, that probably means that we need to look at algorithms that are kind of modular in a way that you can take what you learned in one domain and somehow transfer it to another domain. And so my question to you is just to what extent can we also take observations seriously that come actually more from the behavioral side and what brains can do, and how can this constrain our models?
And something I think I know it best from the work of Josh Tenenbaum, who has been saying this for a long time, right? We are like artificial neural networks drivers in the wrong corner. We should really understand how humans can learn something from one example or few examples. And so he argues that learning takes place on several levels of abstraction simultaneously, right? That there are priors formed for lower level networks. And so this is not addressed here at all. There was one kind of a prior somewhere in there, but one could think of this of maybe being kind of hierarchical prior even, or kind of which is not drops from the sky, but is maybe also learned on a more slower time scale. Or also innate knowledge, I think, is really, an important question right now. How can innate knowledge, something that you put into the network, really enable it to learn certain things very fast? And I fully agree. I think one has to take data, also behavioral data, seriously, right? I think not. Otherwise, we are easily as theoreticians. We have our favorite games then, right? And we are carried away by them if nobody pulls us back.
I like math as much as the next guy, but I mean, you have to know something about the brain to triage. So, for example, one-shot learning, you need a hippocampus or amygdala. If you don't have those, you can't do the one-shot learning. And there's a beautiful recent paper on the, I think it's a rat running on a treadmill, and it turns out the rat has a different thalamic bus for visual input that it can predict that's consistent with its motion, and another bus if it's inconsistent. So the idea of that at some very high level, the rat divides the world into things that are consistent with its internal state and things that are inconsistent. And I think it's very important to have these large scale dichotomies on the table to situate the math in the right place. Otherwise, if we think math is just going to sort of spread like a virus through the whole brain, I think we'll get into trouble.
And I think more general, right now you mentioned this hippocampus is doing certain things, right? In theory, we always look at a generic network of neurons, right? We don't even think about where it is in the brain then, right? And so this is, I think, really a defect, right? There are specialist brain networks, right? And hippocampus is a special one where one knows a lot about. And typically, at least coming from abstract theories, so we ignore this, right? Because maybe it hasn't been kind of been made salient enough and concise enough for us. What are kind of difference in the computational goals, in the circuit organization, in learning rules in different areas then. But I think it's certainly kind of a skeleton in the closet that we always want to look at this generic networks of neurons. And I think it's problematic. Yeah.
About the drift of the neural code, I think the examples you show is a remapping phenomena, right? Where it's not technically a drift, but it's just an abrupt change of the sequence because it's in a different environment, or it's in the next day. So like some contact signal might change, so that causes.
So you're talking about this data from the CIF lab in Stanford?
Just the data you showed.
Yeah, so there were, I think, not two of here, but there, yeah. So this is from the CIF lab then, yeah. As far as I remember then from the paper and his talk then, I think they tried not to do anything drastic to the rodents right now. They were just put in the home cage and then put back every day on this linear track then. So of course, there are other things, they learned things, right? And it could be, this is the result that they learned in the same time. Probably they learned other mazes then, right? Or the home cage, they have a separate map then, right? And so they're kind of interfering then.
Yeah, so I'm just, because I think that it might be just due to, because you put it in another day, so a lot of things happen. So the state of the network might have changed a lot. And
I think one can turn this around also, because when we look at network learning in models or in theory, we always assume this thing has to learn one thing and nothing else then, right? So you could also say this is simply a kind of consequence of having to learn many different things and maintain memories and update memories of many different things, right? And so then you have interference of these memory traces then.
Yeah. I'm glad you put that up, because I wanted to ask you about this before and you've moved on. This cell number 1, fires early on the track. There's a track here, right? That's what it is. So early in the track, he fires and then stops firing. So this cell has a place field early in the track on this day, 5. And that point on the track at day 10 no longer activates that cell, nor at 15, 20, 25, etc. So when I said, I was talking about this as a memory, this cell is a cell that responds to the cues that somehow indicate that it's early in the track, early in the, on day 5, and later it no longer responds to those cues. So it's as if it's like a little recognizing unit that has forgotten this pattern. That's how I interpret it. I mean, this is basically, what's interesting about this is that these cells, as you look along here, there's hardly any activity in those cells on this track at all.
And I think this may be kind of an artifact of plotting, because you see here, here they have reordered it according to the order in which they fire here on this day then, right? And so here it looks much more activity here than here then. So this is just something which I think just comes from this color code then. So
it forgot its feature, but it learned some other features. I mean, it really seems to be latched onto some other features.
Right, it's just turning up, but that's kind of what an episodic memory would do if it has a limited capacity. It fills up and it moves up.
So just to understand, Wolfgang, are those the same neurons that day 15 is day 5? In this
this is what they argue in this paper.
No, in the second column, right? Sorry.
It's not the same set.
These are the same neurons. I think actually, there are more neurons in, right? So let's see.
I think it may be a different animal or a different.
Right, no.
This is a different number of neurons.
Yeah, so this I didn't notice. Sorry about this. Yeah, ordered for day 5.
I think you can order them if you go to day 20, you can order the cells such that they fire in the order that the track, that the animal runs along the track. So this happened to be the cell that fired first along the track, this is the second, third, on day 20. And then if you look at those exact same cells 20 days ago, they were not organized in the same way on the track, or firing at all, it looks like here.
Well, it looks like if they're the same cells, they were firing, you just can't see it, right? They're just in a random order where you just can't see it.
Yeah, they could be, or they may not, those cells may not fire along the track.
So that's the question, are these the same cells or not, or they're just in a different order?
Yeah, I think at least these are all the same cells then, right? And these are all the same, right? But I agree, it's something, sorry, I didn't look at this then, And so here, this is at least, I think these are the same cells. And maybe they do a certain selection that they're here selecting out of much larger pools of cells, those neurons which fire at a particular time on this day, right? And so this defines now this 222 neurons then.
Sure.
And possibly, from the same pool they draw these things then, it's just a different mapping, but we have to look at the paper,
It's getting a little bit past the time. Maybe we should break and have people continue outside. Thank you, Wolfgang.